guys, this is Jen here, and today I'm going to show you how to make an edible DNA model. And what you'll need for this project are 2 Twizzlers, about 9 to 10 toothpicks, and some mini rainbow marshmallows. And you'll need about 9 pink ones, 9 green ones, 9 yellow ones, and 9 orange ones. Okay, so DNA is the foundation of all living things in plants, animals, and humans. It contains a set of instructions needed to make proteins and molecules for our bodies so that it can grow, develop, reproduce, and survive. Now, DNA has 3 parts. Let me show you these 2 Twizzlers here that I've chosen with different colors. Oh, here we go. Okay, so these Twizzlers here represent the backbone of DNA. This red one represents sugars, and this green one here represents phosphates. Now, sugars and phosphates are very important for our bodies as they help store and transfer energy to our bodies. And they're also both water soluble, which means they can easily dissolve in water, which helps to transfer and store energy from the sugars and phosphates. These form the outer structures of the DNA. And the third part of DNA are these marshmallows here. These 4 are called nitrogen bases, and they each have their own name. For instance, this pink marshmallows are called adenine. The green ones are thymine. Yellow ones are cytosine. You can see that pretty good here. And the orange ones are guanine. And these are not water soluble, so that means they can't really easily dissolve into water like the sugars and the phosphates. So what I'm going to do is I'm going to take this toothpick and I'm going to use that to attach my nitrogen bases in pairs to the Twizzlers. So let me just take a pink one. This is thymine. And I'm going to take the green one. No, pink one is adenine, the green is thymine. So I'm just going to take this Twizzler. And this toothpick represents hydrogen bonds. They help the nitrogen bases to chemically link to the outer structures of the DNA so that it can form into nucleotides, which are the building blocks of DNA. And once they're formed, then they become water soluble to where they're able to transfer and store energy through the water. So let me just go ahead and take my thymine and just attach it to the toothpick just like that. And then I'm going to attach that to the Twizzler. And you want to be careful because these toothpicks are very sharp, so you don't want to poke yourself. Then I'm going to take this green marshmallow, the thymine, and attach it to my adenine. See? Just like that. Okay. Next, I'm going to take a different pair. I'm going to take this yellow and orange. Now this yellow one, if you remember, represents the cytosine, and this orange one represents the guanine. So I'm just going to pair it up just like that, just like I did with my first one. Attach it to my Twizzler. I give it a little more room. There we go. And I'm going to attach my orange one, the guanine, to my cytosine. Oh, it's also important to note when it comes to nitrogen base pairing is they have very strict pairing rules. For instance, the adenine, the pink one, always pairs up with the thymines, the green ones, just like the cytosine always pairs up with the guanines. And the reason the pairing is so important is so that it's able to process and read the instructions so that the DNA can function correctly. And you just keep going down the line. I'll give you more examples. take a green thymine, attach it to my adenine after I attach it to the Twizzler. Now, the sequence of the pairing, I mean, as far as going down the line, really doesn't matter at this point. So you can just get as creative as you want to, just as long as you remember the base pairing rules for the adenine, thymine, cytosine, and guanine. Just go ahead and take my orange one, my guanine, and to pair it up like that with my yellow cytosine. And you just keep going down the line, and then once you're finished with that, you just attach it to the other Twizzler. And you should get something that looks like this copy right here. And then you're ready to move on to the final step, which is forming the double helix, also known as the twisted ladder. And in order to do that, you just hold on to this like that. Use your top fingers to hold onto the top side and the other fingers on the bottom, and you just kind of twist it like that. See how it's twisted? You can get a better view. Yeah. You twist it just like that. And what's the cool thing about this is the reason the DNA twists like that is so that it can replicate and make copies. And it also helps to keep water out from the empty spaces here so that it won't disrupt the process. And that is how you make the DNA model. I hope you like it, and I'll see you next time. Bye.